right. Well, I'm acutely aware that I'm standing between you and lunch, so hopefully this will go quickly and smoothly and we'll all be able to have dinner. Can you hear me at the back, John? It's okay? Great. So I first need to acknowledge my co-authors on this presentation. They have done some amazing work over the past 12 years that we've been working together. We've had students involved, we've had postdocs involved, and the people I've highlighted here have really been the drivers of the figures and the work that I'm going to show you today. But there's a myriad of people behind them that set this stage. And the first time that I was invited to present at FRI, I think I presented the promise of what genomics could do for forestry, and particularly for sustainability, for resilience. And what I tried to do today was make good on that promise and show you where we've made the most progress, where we've actually been able to deliver outcomes that are useful to the industry, to government, and to other stakeholders. And so this team really has bought into this notion of making genomics useful for your applications. So why do we get so excited about genomics? Well, particularly in the forestry realm, genomics has enabled so many things that we could only dream of maybe 10 or 15 years ago. And it's because we can do high-throughput, precision analyses. So if you think about being able to do meaningful sizes of samples that we can then make decisions or make tools that are actually effective in any sort of stand or landscape, we need to do a lot of samples. And if we want to tell the difference between things that perform similarly, we need to be able to drill down into what exactly makes them different. And so this is what genomics has allowed us to do. And when I was standing up here 10 years ago and talking about genomics, we had a few hundred markers, we had a few thousand genes, and now we have thousands of markers, and we have even more of that, of genes. So that has really unleashed our ability to tackle the problems that I'm going to talk about today. So being able to monitor a whole bunch of sequences all at the same time, being able to monitor hundreds or even thousands of individuals all at the same time, and being able to measure not only those genes, but what those genes are doing. So actually getting under the hood of these plants, in our case, we're talking about the trees, and asking what they want. So instead of me guessing and going after my favorite trait, we can lift the hood and say, what is going on inside of you to the plant? And so I'm going to cover a few species today, but we actually have genomic resources for a lot of the species that are germane to Alberta, to BC, to Saskatchewan. And not only do we have those resources for the trees, but also the interactors with those trees. So Justine has talked about some of these guys. We've talked about some of the beasts. Dazine talked yesterday about some of the beasts that take these on. Having the resources available, not only for the trees, but for the organisms that interact with them, enable us to do our work as well. So for those of you who may have attended a workshop that we held a couple of years ago on genomics, this list will not look any different from what we covered in an entire day. We're not going to cover all of these because it'll be about 2 minutes per. Andreas has expertly covered seed zones, and so I wouldn't want to add anything to what he was bringing us in his presentation. And just on the sake of time, I'm not going to cover the last few topics there, but at lunchtime, I'm happy to provide anybody who's interested in these topics with concrete examples of projects that are going on here in Alberta and other places in Western Canada that are delivering genomic applications in these areas. So I'm going to cover those middle four, starting off with risk assessment. And this is building off of the talks that we had during the TreeNet session yesterday, focusing on the trees in our particular case, and how we've used genomic data. I'm always afraid of pressing the black button of death on here. the genomic data from our trees and their interacting partners in spatial mapping and in forming model development that then has been used by our stakeholders and our end users in all different kinds of ways. And what I'm going to do today is just build a little bit more on the spatial mapping side. And this has been led principally by Kathy Cullingham, who was previously with TRIA, is now involved in a different project, but she just loves this so much. She's even here today. So thank you, Kathy, for showing up. So one of the examples that we go back to time and again are the markers that Kathy developed to distinguish lodgepole pine from jack pine and the hybrids. And so these markers are available today downtown at Delta Genomics for anybody here to use on their own volition if there's any question of the ancestry of the genetic stock that you're working with. And if anybody's interested in accessing those resources, I'm more than happy to provide Michelle Miller's contact information, and she's more than happy to help you out. Where this became important is through the work, the diligence of Tom Hutchinson in identifying those jack pine, and they were jack pine, that had been successfully attacked by mountain pine beetle back a few years ago. But now we have a spatial layer that's recently been updated by another student involved in TRIANet that provides us with a spatial understanding of what that hybrid zone looks like and the complexity of that hybrid zone. That these are not just simple margins, but within that hybrid zone, we have a true mosaic. And we know that because of DNA markers. So Kathy's built on that early success, dove in with a few hundred more markers to look at whether or not we could use these DNA markers as an indicator of being able to distinguish the experienced region of lodgepole pine, that is the region of lodgepole pine that had previously experienced mountain pine beetle. Could we distinguish that from the naive region? So we can do that with climatic suitability index, but could we refine it? Could we add a genetic component that could allow us to develop a more precise layer? I'm giving you one example here of one particular gene that shows a signature of positive selection across this hybrid zone. And this particular beast is actually involved in phenolic targeting. This is work that recently came out of one of my favorite colleagues' labs in Belgium. And this guy responds to the fungal inoculation with Grossmannia clavigera in jack pine, but not in lodgepole pine. So now we're starting to get the genetics, and this is, I want to thank Andreas for giving us a shout out on the landscape genomics side. Not only are we getting these spatial maps that maybe we can start to use to inform the risk assessment framework for mountain pine beetle, but we can start maybe to get to adaptive features of these populations that might be affecting susceptibility. So Kate has built on this work that was carried out in this more local area over a national scale, and that work is ongoing, trying to understand whether any of these populations of lodgepole pine or jack pine might be more or less susceptible to mountain pine beetle and the fungi that they carry, and also to some of the other drivers that we think impact population dynamics like drought. So we think about other risks. We can have risks, as we've talked about mountain pine beetle lots in this meeting, but we can have risks from other pests and other pathogens, hopefully not being transported across borders and raw logs or anything like that. But if they were, there are a number of genomic tools that have been developed, principally by Richard Amelin's group, to detect those beasts should they arrive in an unanticipated location. And so these are based off of having the genomic resources to have those species diagnostic markers. So these are extremely useful, particularly when it comes to interfacing with agencies like CFIA. Our interest is in using these tools to monitor the pest or pathogen in situ, so in wood, in leaves, and sometimes these markers let us down because they are meant to discriminate species, but maybe not sensitive enough or maybe not specific enough to work in wood, for example. And so we've gone on to develop a set of markers that can be used for the ophiostomatoid fungi that are carried with mountain pine beetle. And if anybody has any questions on how we've used that, I'm happy to answer them over lunchtime. So one of the areas where we can address sustainability and particularly resistance within the framework that Darren has laid out, within the framework that Erica laid out yesterday, this notion that we have to prepare for tomorrow today, and there's room within the policy framework for putting genetically improved trees, or at least selected trees, on the landscape is tree improvement. And this is probably, if we think on the international scale, where genomics has been applied the most in forestry. So these are traits that we are involved in looking at, and some of our collaborators are involved in looking at, and also other projects going on, say, for example, in BC and in Alberta. And so why do we even care about bringing genomics into tree selection? Isn't it just better to walk the woods and find those better trees ourselves? Well, it's because tree breeding programs, as several of you know, are not for the faint of heart. It takes about one career to make this loop, maybe longer here in Alberta. But where we can shorten that loop up is testing projects. This can take us 25 years or more to get from that selection process right through to collecting seeds, getting those seed orchards going, and then launching another generation. We're impatient, we're in the 21st century, so we can shorten this testing process up through genomics. And that's using the genotyping tools, the DNA marker tools, but also getting to more innovative phenotyping tools. So some estimates would say that we can take that period of 15 years, sometimes it can take, to get to accurate assessment of your genetic stock, because you might need to wait for mature wood, could even be longer than that. We can get that down to 1 or 2 years. So we can start now talking about how do we want to implement these things. This is one project that I'm involved with. The majority of the investigators with this project are in Quebec. This is the project with respect to using genomics for tree improvement that's probably the most advanced in Canada. They draw a lot of inspiration from the work that's gone on in the southeast of the US. In this project, we are using a number of different phenotyping tools to look at a whole host of growth and productivity traits, as well as pest resilience. We have a genotyping tool that we developed a few years back. All of those trees are already genotyped. And then we bring in the quantitative geneticists who do amazing work with these vast matrices of numbers. I don't do that work, I don't do math in public, but what comes out of here are these marker panels and genomic selection models that then we can apply to those tree improvement programs. So within Spruce Up, which also operates with a complementary project called Fast Track, the first proof of principle for this is being enacted now. So this is not future stuff, this is now stuff. And so we've used inspiration from this project to partner with Alberta Agriculture and Forestry on a separate project on Western gall rust. Erica mentioned that yesterday. There are others also looking at Western gall rust. We're using this framework to determine whether we can actually use genomic selection with the genetic material that was made available to us here in Alberta. And based on the phenotyping data that Chandra in my lab has recently produced, I believe that we're in excellent shape. That is the most outstanding data set that I have ever seen for a genomic selection experiment. It rocks. So stay tuned for that one. So this next slide will probably take a really long time to come up. There we go. So we heard from Darren that the shoulder seasons might matter in our context of climate change. So within Spruce Up, one of our focuses, or foci, has been phenology traits. So where we see these gray parts are chromosomes, and where we see these bars, those bars are the genomic regions that are controlling these traits. And so we've got genomic regions now. We've got genes under those genomic regions. We've got markers within those genes. This is the information that is being transferred to the end partners in the ministry and in the province of Quebec, and also Irving for their breeding programs. So I'm going to switch gears a little bit to how can we use natural populations in tree improvement. And I want to talk about host quality in mountain pine beetle. So this picture is taken in Norman Lake. Staff in Lindgren, when I visited a few years back, took me on a bit of a trip. This is about 5 minutes south of my brother's place, as it happens. And I'm not sure if in the light you can see it, but there's a couple of green trees in amongst this sea of destruction. So these survivors, are they lucky? Did they just escape being detected by random chance alone? Was there an attempted attack that they were able to rebuff? Or is there a genetic basis for why they survived when all of these other guys were victimized? And so through our collaboration and through our collaboration with Dizzy and Huber, and Dizzy is here, as you know, we embarked on a genetic analysis of the population that they developed from seed that was collected off of the dead trees in these stands, along with seed that was collected off of the living trees. So we've got our killed trees, and we've got our survivor trees, killed trees, survivor trees. And so the killed trees, their cones would have been set before they died. And so they could have been fertilized with pollen that could come from dead trees or survivors. We don't know that pollen source. But what we do know with the survivors, we can actually, because lodgepole pine is holding its cones on the branches, we can actually distinguish two types of cones. Those cones that were produced before the outbreak and cones that were produced after the outbreak. And so the cones that were produced before the outbreak, we know that mom's a survivor, but dad could have been a survivor or a victim. But for these guys, these cones that were set after the outbreak, both those parents had to be survivors. So this is work that Kate has been doing with Bianca, who's an undergraduate at my lab, who's just finished her last exam, so we should all give a cheer for Bianca that she's moving on to the next phase of her life. We can see genetically that we can distinguish the dead trees from the survivor trees, or at least the progeny that come from dead trees and survivor trees. So genetically, there's some differentiation there. We're excited by that. So Bianca took a very rough first pass for her undergraduate project in seeing whether we could identify markers that associate with surviving versus being victimized. And this is our very first pass using a general linear model approach. It's not the ideal approach for this, but it was the quickest approach, so we've started there. We can move on to more complex models that take into account a few variables that we should acknowledge. But what we're really encouraged by is there are markers that are exceeding our threshold of differences in frequency between the victims and the survivors. So this gives us confidence that there is a genetic basis to being a survivor on the landscape, even if that was a massive, massive outbreak that was experienced in those regions. So super excited by that. I want to switch gears now to another topic that relates to genetic detection on the landscape, and this is traceability. This is an issue when you think about the reforestation chain for many of us in the room. We start out with cone collection, shooting branches off trees. Cones have to be brought in. Those cones need to be dried. Seeds need to be sorted. They need to be accessioned. And then we've got withdrawals, and we've got seedling production. And finally, those trees get in the hands of the planter, and they go out in the field. But anywhere along that line, there's a possibility for a mix-up. So can you trace your genetic stock? So for many years, we've been able to trace clonal material. That's not been a problem. We've developed markers for derrick sitters, for example, that could be used to distinguish different clones of willows in a still beds. But what about the case where we have genetic material from many different parents and genetically unrelated going out onto the landscape? So we embarked on this test case, which we call CSI Rhiannon, because Rhiannon has done all of this work, and it's going to be up to her to answer the questions because she does the complicated math that I don't do. But if we had a mix-up, could we use the genomic markers that we have on hand to distinguish these accessions? And can we discriminate between seed lots that are actually physically close together? So here I need to give a shout out to AdaptTree. It's led by Sally Aiken at UBC, involves Andreas Haumann. I'm super impressed by their collegiality in providing us with their SNP chip in advance of publication that we could use in this project and the previous projects that I mentioned. That kind of community collegiality is essential for us to be able to deliver and make good on our promises to you to make genomics useful to the community. So we have these 8 seed lots, mysteriously numbered 1 through 8. This is a location in Alberta. A couple are from orchard materials, so improved materials. They're obviously not on the map because they come from different areas. And so this guy is marked as 6 here, but he's actually 7. And so can we tell? Can we tell whether this guy is actually from where we think it should be from, or was there a mix-up? Maybe it came from one of the other areas. So we have 3 lines of evidence to say that this unknown actually is this number 7. The first line is something called Nay's FST. This is the same kind of measure that we saw in Bianca's work, a measure of differentiation. And so we see that 6 and 7 land close together in this measure of genetic differentiation. We also see that outgroup, the one from southern Alberta, easily distinguished. These other guys are pretty close, genetically speaking. But we see that these guys are the closest. So that's our first line of evidence. Second line of evidence is this idea of using a genetic assignment test, which is meant to take unknowns and see who they're closest to amongst your knowns. And so in our assignment test, which is called structure, we see that 6 most aligns with 7. So here we've been another line of evidence that this unknown seed lot is actually this seed lot number 7 and not any of these other guys. Third line of evidence is a relatedness test, kinship test. So this is where we're eternally grateful that we've got people in a department like Dave that work on furry things that happen to be promiscuous, and so they know all about these relatedness tests. And so here we have that third line of evidence that 6 and 7 show this high level of relatedness, or at least some level of relatedness, relative to all of these other groupings. So collectively, this allowed us to, in this case study, to provide solid evidence that the unknown was in fact what we thought it was, which was that seed zone 6. So I've given you a few vignettes here of how we've been able to use genomics to address challenges that are faced by the industry, that are also faced by government agencies and others in addressing questions related to resiliency and addressing questions about how do we make sure that forestry today continues to produce resilient landscapes and continues to support the companies that operate within our boundaries. One can imagine that we're just getting started, and there are other applications that can come out of this to continue as the industry evolves We heard about LiDAR today. Maybe there's going to be more remote sensing. If we can think about precision agriculture, can we think about precision forestry, particularly if this province follows leads of other provinces, and maybe we get into more of a triad scheme or something like that. This can also involve biotechnology, but it doesn't have to. And so that's something that comes onto the table as well. And so with that, I have to thank a whole lot of people, particularly our collaborators and people in my lab, past and present, who do all the hard work on this stuff. And then lastly, I need to acknowledge this man. This is my dad. My dad spent his entire working career in forestry, in and around Prince George. Amongst the other things that he did was salvage logging following the spruce beetle outbreak in the early 70s in what is now McGregor Model Forest. Other kids went to Y camp. I went to logging camp. My dad passed away last month, and I'd like to dedicate this talk to him. Thank you.